Michel Sadelain is director of the Center of Cell Engineering and the incumbent of the Stephen and Barbara Friedman Chair at Memorial Sloan-Kettering Cancer Center. He's a member of the Immunology program and the Departments of Medicine and Pediatrics. Dr. Sadelain's laboratory has made several seminal contributions to the field of chimeric antigen receptors, CARs, from their conceptualization and optimization to their clinical translation for cancer immunotherapy. His group was the first to publish dramatic molecular remissions in patients with chemorefractory acute lymphoblastic leukemia following treatment with autologous CD19 targeted T cells. So we'll start with Michel. So Dr. Sadelain is joining us from New York this morning. So welcome, Michel.
Thank you, Janet. First, I'd like to thank you and the Gairdner Foundation for the invitation to this very timely and very interesting symposium. Let's see, you can see here my disclosures. CAR T cells are born in the thymus, where they not only acquire the letter T, but also a T cell receptor, a clonotypic receptor specific for a given antigen HLA peptide complex. We create a vast repertoire of those T cells, enabling us to be protected against a number of foreign invaders, such as viruses and sometimes cancers. But this repertoire, however, is not infinite. In fact, before T cells were the source of T cells was even uncovered, scientists had begun to remove lymphoid cells from one animal and transfer them to another to assess and study their function, coining the term of adoptive transfer. As early as the 80s, this inspired a series of clinical strategies to treat cancer that were all based on the adoptive transfer of blood cells, leading to what was called adoptive therapies. These included donor leukocyte infusions, the infusion of cells that had been cultured and activated with lymphokines, the removal of T cells from tumors and their expansion prior to their reinfusion to the patient, known as TIL therapy, and also the use of virus-specific T cells, particularly in the post-transplant setting. Collectively, this vast endeavor spanning several decades have taught us that T cells can be very effective. They can also be dangerous. To be effective, however, it was clear that one needed to enrich these T cells in T cells that would be actually specific for any given cancer. And so was born this idea that perhaps we could instruct the immune system, genetically instruct the system, to recognize through its T cells any target or antigen of our choosing, which is found on a cancer. And so was born this idea that led to what is called today CAR T cell therapy. This therapy is at once a gene therapy, a cell therapy, and an immunotherapy. It draws very much on fundamental immunology as well as tumor immunology. But it is indeed heavily predicated on genetic engineering, starting years ago with the use of retroviral vectors, and today moving towards genome editing using Crispr and other tools. But furthermore, it also makes use of a novel class of receptors for antigen, synthetic receptors, that some years ago we termed chimeric antigen receptors. These receptors don't recognize HLA peptide complexes, as does the physiological receptor found in T cells, but rather cell surface structures, and for which a molecule called CD19 has now become really the paradigm and poster child. To advance preclinical research and convince ourselves and the world that this approach had any merit, we also had to advance cell manufacturing sciences, which really developed at a few academic centers, mostly in North America. And to bring this to trial, of course, one had to fit the manufacturing process within regulation set forth in the US by the Food and Drug Administration. So the work starts, at least for me, in the late 80s, after leaving actually the University of Alberta, going to MIT, wanting to learn the rudiments at the time of gene transfer in mammalian cells. And it took several years to figure out how to introduce genes into, at first, mouse T cells using gamma retroviral vectors. And today we have several other tools consisting in particular in what are called lentiviral vectors or DNA transposons that are all efficient at transferring genes into T cells. One also had to develop these receptors that I already alluded to, and there's no time today to review their history, but suffice it to say that the physiological receptor, the TCR and CD3 complex, is made up of multiple chains. And one chain in particular, called the CD3 zeta chain, is particularly important in initiate the process of T cell activation when a T cell encounters the antigen, which it is able to recognize. And so following the cloning of this chain, certain fusions of the CD3 zeta chain were created to impart upon these fusions the ability to recognize cell surface molecules. Owing to the ability to study these fusion molecules not only in hybridomas or clones, but in actual real T cells, if I can say primary T cells, we realized that zeta chain fusions were not capable of directing a productive response, but they could do so if we introduced two signaling motifs in these molecules, which we eventually called second generation CARs. Now, to study this in the laboratory and eventually in the clinic, we also had to identify a target. And in the late 90s, we set our eyes on a molecule called CD19, which is found on the surface of leukemias and lymphomas. As it is on the surface, it can be engaged by CAR T cells. It is also found in some normal cells in the body called B cells, and we therefore anticipated that CAR T cells might eliminate normal B cells owing to their expression of CD19, but that this in the clinic could be acceptable. And so as you see here, it's almost 20 years since we provided the first demonstration that one could take human peripheral blood T cells, transduce them with a retroviral vector, encoding the gene or the cDNA, producing the CAR molecule, and that then, following a single infusion of those CAR T cells to a mouse bearing a CD19 positive leukemia or lymphoma, that these mice could be treated and even cured. This now had to move to the clinic, and here I would like to pay tribute to manufacturers at academic centers who greatly contributed to advance this field. Carl June, some years before, had developed some beads coated with antibodies to CD3 and CD28, which could be used for the expansion of T cells under clinical or cGMP conditions. And here are the three leaders of the three centers that produced the first CD19 CAR therapies: Bruce Levine at the University of Pennsylvania, Mark Dudley at the National Cancer Institute, and my colleague at Memorial Sloan-Kettering, Dr. Isabelle Rivière, who set up a facility designed to manufacture CAR T cells, as well as this manufacturing scheme that was soon approved for use in phase 1 clinical trials at MSK. The first trials focused on patients who had relapsed and refractory B-cell malignancies, either leukemias or lymphomas. And the first reports were very striking. The NCI reported on a first patient with non-Hodgkin lymphoma who went into a complete remission following a single infusion of CD19 CAR T cells. The University of Pennsylvania was the first to report on 3 patients with chronic lymphocytic leukemia who also showed very profound responses to this treatment. And we were the first to report on the first 5 adults with acute lymphoplastic leukemia, showing, as you see in the lower left panel here, that following the infusion of these CAR T cells, this cloud on the right of CD19 positive leukemic blasts is completely obliterated. And as you see at several time points in the weeks that follow the infusion of these CAR T cells, these tumors are undetectable. These responses qualified not only as complete responses, but molecular responses. Later that year, the journal Science proclaimed cancer immunotherapy to be the scientific breakthrough of the year. This owed largely to a more advanced form of immunotherapy that makes use of antibodies to unleash latent T cell responses. But the editors also saluted this emerging immunotherapy based on genetic engineering. And in subsequent years, those three original centers and others confirmed these results in larger trials. It's in the course of a conversation with a journalist who asked, well, what is it exactly that you do? This term came out of living drugs. What do you do? Well, we create living drugs. Indeed, the T cell that is armed now with the CAR, upon its infusion to the cancer patient, will find the tumor, hopefully kill the tumor cells, but also multiply. And so the army of T cells fighting the cancer expands in the body of the patient. In a nutshell, here is where the field stands today, from showing you here 4 large studies with already some more significant follow-up, following again the single infusion of CD19 CAR T cells with patients who either had refractory leukemia or lymphoma. On the top left, our own studies in adults with refractory ALL. What this slide does not show is that about 85% of these patients who had no longer any therapeutic option went into a complete remission. You see, however, that half, and even slightly more than half, will eventually relapse over time. And yet, a sizable fraction will survive long-term, and hopefully we will soon declare them to be really cured of their disease. To the right is a study from the Children's Hospital of Philadelphia that Kathy High comes from, where the first study treating children with ALL was conducted. And there too, a remarkable complete remission rate on the order of 85 to 90%, but still you see about half of the patients relapsing over time. Some centers make use of a CD19 CAR that uses in blue here a CD28 costimulatory domain, and others a 4-1BB costimulatory domain. But as you can see, the results are overall similar. And below are 2 studies in non-Hodgkin lymphoma. One multi-center study on the left using a CD28-based CD19 CAR, and one on the right from the University of Pennsylvania using a 4-1BB CAR. In lymphoma, the complete remission rate is not quite as high as in the leukemias, but on the other hand, the responses are durable in slightly more than half of the patients. So clearly dramatic results in patients who otherwise would not have survived really more than a few months. But on the other hand, clearly room for improvement. In 2017, the FDA approved the first CAR therapies. Both of them are specific for CD19. Both of them are second-generation CARs, and one utilizes the CD28-based CAR, and the other one, this 4-1BB-based CAR, which was originally developed by Dario Campana at St. Jude Children's Research Hospital in Memphis, and then superbly developed at the University of Pennsylvania and at CHOP. These approvals come with a limitation or a restriction called REMS. The REMS program means that these therapies cannot be delivered just at any center. The investigators at the center who deliver this therapy have to be appropriately trained and have to report any adverse events. Following the approval of these therapies, there was a very dramatic, again, interest in advancing cell therapies really worldwide. As you can see, there are today over 500 clinical trials listed at the clinicaltrials.gov website. About a year ago, it was estimated that at least 20,000 patients worldwide had already received CAR T cells, and also that about 145 biotech companies or large pharmaceutical companies had a CAR program. It is also noteworthy that about 90% of all these trials take place in the US or in China, with actually China leading the US in this realm. The three impacts of CD19 CAR therapy at a high level really are that, first and foremost, they provide hope for patients with refractory B-cell malignancies. But they also mark the first FDA approval of an engineered cell, genetically engineered cell, as a drug. And thirdly, this is really the first cell therapy that swayed the pharmaceutical industry to enter the world of cell-based medicines. Now, there are side effects and there are, of course, failures of this therapy, and we learned a lot about that in the recent years through all of these clinical trials. In addition to the elimination of normal B cells that I already mentioned, two other not anticipated toxicities have been observed in patients undergoing CAR T cell therapy. One is called cytokine release syndrome, and as the name suggests, is the consequence of the large production of cytokines in the patients at the time that these T cells are fighting the cancer. The second one is a neurotoxicity that originally was thought to be part of this CRS syndrome, but which we know today is a separate entity, although yet poorly understood. In terms of the CRS, though, we have made progress, and recently some mouse models have been established. And the first lesson really is that one of the essential molecules that mediates the toxicity of CAR T cells, which is interleukin-6, which can be produced by T cells, is in fact largely produced by myeloid cells, in particular macrophages that are very commonly present at the tumor site. And this today is informing additional and new approaches to attempt to prevent the CRS and not just react to it once it develops. On the right here, I've listed some mechanisms of resistance or failure, if you like, of CAR therapy, and broadly they fall in three categories. One is the defective T cell function. The quality of the cell may not support the therapy, perhaps because the patient did not have good cells at the beginning, but also because the engineering process may have weakened, if you like, the T cell. This obviously is the emphasis for much research going on today. The second is microenvironmental suppression. As the CAR T cell enters the tumor site, it may be inhibited by a number of actors found in the tumor microenvironment. This applies to any immunotherapy, not just CAR therapy, but those that I list here have been directly shown to indeed dampen the activity of CAR T cells. And this too leads today to a number of strategies aiming to overcome these specific barriers. And the third one, which is also a hot topic in the field at this time, is referred to as antigen escape. Indeed, if you target one molecule, CD19, for example, what would happen if some of the cancer cells become CD19 negative? Well, they would escape. And so there's great interest in defining strategies that can mitigate this escape. And so these failures, our limitations, are essential to us, at least on the research side. They provide to us the roadmap of what we need to do next to improve this therapy. And I would like to give you just a quick few insights into that, just to show you perhaps where the field is going. And so there is, at least at my center, research ongoing to identify novel targets, to, of course, improve the T cell engineering per se, using the CAR T cell not as only as a killer of tumor cells, but what we sometimes call a micropharmacy, meaning that the T cell, once it has entered the tumor microenvironment, may be used to release additional molecules that can still enhance the activity of the immune system against the tumor. Then there are combination therapies, For example, checkpoint blockade that I already mentioned together with CAR T cells, and one that I will mention quickly in the next slides, whereby CAR T cells are also combined with radiation therapy. And fourthly, shown below here, is a really worldwide effort to look into other cell sources other than T cells collected from the patient himself or herself that could be used for the purpose of such therapies. And I'll say something about that quickly at the end. So some examples of how this field is evolving. Well, again, on the upper left here, you see the CD28-based CAR. The green domain here is that zeta chain borrowed from this T cell receptor CD3 complex that you see here. It is fused to the signaling domain of this CD28 costimulatory receptor. As you can guess from this drawing, the function of the CAR doesn't quite replicate the way things would work in the context of a normal T cell response. Typically, there is a 3-to-1 ratio between CD28 and CD3 molecules. And also, the CD28 molecules enters the synapse that forms between the T cell and its target with a slight delay. That's not the case in the CAR, where everything is already packaged at a fixed stoichiometry of 1-to-1, ready to go. And in fact, observing results in patients and noticing the limited survival of these T cells, even when they produced deep responses and complete responses, we started thinking that maybe this signal provided by the CAR was too strong. And so we started mutating some domains known as ITAMs that immunologists know well that condition the strength of activation provided through the physiological receptor. And without going into details, we found that as we diminish the strength of activation, we can create some CARs that are worse than the original and FDA-approved CAR, but then some that are better. And in particular, this one called 1XX has now become our favorite. And I'm pleased to say that we've just treated our first patients at Memorial Sloan-Kettering using T cells engineered with the 1XX CAR. The main property of this CAR is that it maintains strong effector functions, but allows the cells to survive much longer, thus giving him more time, if you like, to fight the cancer. Another example of advance in the field owes much to genome editing. And last month, the Nobel Committee rewarded the work of Jennifer Doudna and Emmanuel Charpentier, their magnificent work on Crispr. This is a tool that allows all of us now to enact precision engineering. And this enabled us to ask the question, when we introduce the CAR gene into a T cell, does it make a difference where this gene integrates? When you use a gamma retroviral vector or a lentiviral vector or a DNA transposon, all of which are effective, the gene integrates in a different location in each and every other cell. And this results in what is called variegated expression of the gene, which means it's not quite the same in each and every T cell, and also can have adverse consequences owing to insertional mutagenesis. So the advent of a tool such as CRISPR allowed us to ask, well, are there some locations that we could choose and some that are better than others from which to express the CAR cDNA? And so in human peripheral blood T cells, we took again this FDA-approved CD1928Z CAR and introduced it at different locations, and one in particular called the TRAC locus, which encodes the alpha chain of the T cell receptor. And as you can quickly see from these studies, these mouse studies below, these are what we call CAR stress tests, in which we infuse very low doses of T cells to tumor-bearing mice. We can see when comparing a conventionally engineered CAR T cell to an edited CAR T cell, that the latter wins. You see at the same dose, it induces these long-term remission, whereas the classic engineering approach produces a cell which at this dose cannot eliminate the tumor. So why are these T cells better? Well, because they pace themselves. They express the CAR at an optimum level, not too much, not too little. It's internalized after doing its job, which means that the T cell then takes a break and new CAR is translated from RNA and comes back to the surface, allowing the T cell to work again. And I'm pleased to say that Dr. Riviere and her team at Sloan-Kettering are close to opening a trial, hopefully at the beginning of next year, that will make use of CRISPR-edited CAR T cells. And we hope to test the idea that having more efficient T cells allows us to give a lower dose, and that at a lower dose, there will be fewer toxicities. Another example of progress in the field comes from observations such as this one, again taken from our own studies, where we found that combining gamma radiation, low dose radiation, doses that are commonly given to patients in the context of palliative therapy prior to a CAR T cell infusion really synergizes. So here you see in this leukemia model, a low dose of CAR T cells is ineffective, the radiation at this dose is ineffective, but the two together show remarkable activity. And we are studying this, and we and many others around the country now are also planning clinical trials to assess this combination therapy. I also want to mention the need now to identify novel targets. Years ago, we talked to experts in this cancer or another and reviewed the literature. But today there's a need to really more systematically investigate what you could call the surfaceome of tumors. What exactly are all the possible targets that exist on the surface of different cancers? And so we in our lab have engaged now in systematic bioinformatics approaches that largely build on cell surface proteomics and are starting to identify at least candidate targets in different cancers. So where should we apply CAR therapy? Well, this principle could be adopted in the context of many cancers and even, as you'll see shortly, beyond cancer. But here in this slide, I put it in the context of cancer immunotherapy. This slide shows you on the y-axis the mutation burden found in different cancers. A very famous slide seen very often. And in gray here, I've circled those cancers that tend to respond very well to checkpoint blockade. Checkpoint blockade is this amazing immunotherapy based on the infusion of antibodies that release the brakes, if you like, on T cells that are there in many patients, ready to potentially eliminate the tumor, but inhibited by these checkpoints. Now, this therapy works particularly well in patients with a high mutation burden, who are more likely to have a T cell response against those cancers, which I've circled here in gray. But then there's a number of cancers in which the mutation burden is low, and which is the case in ALL, the disease that we initially focused on. And so we think that one particularly attractive area where to apply CAR therapy in oncology is in those cancers with low mutational burdens. So you've guessed that CD19 CAR therapy, at least we believe, are only the tip of the iceberg. And there are many other applications, we believe. First, in other hematological malignancies. And today, there are promising results obtained at several centers targeting multiple myeloma with CAR T cells. Of course, the largest fraction of cancers are solid tumors. All leukemias, lymphomas, and myeloma account for perhaps about 10% of all cancer patients, and many more are solid tumors. I've pointed out earlier some of the directions that I think we can take to adapt the CAR approach to these solid tumors. But then engineered immunity may be useful beyond oncology. One can conceive that engineered T cells could be useful in the context of autoimmunity, intractable infections, or even regenerative medicine. In fact, we recently published an example of that, looking at senescent cells. Senescent cells in the body are normally removed in large part by our immune system, including T cells, natural killer cells, and macrophage. But in some instances, these T cells persist, accumulate, and can lead to chronic inflammation, including ultimately fibrosis and terminal damage to different organs, the liver, the lung, or arterial walls, and others. So using our algorithm to search for targets, we found one target called UPAR present on senescent cells and demonstrated in mouse models of liver fibrosis that using CAR T cells, we could actually remove these senescent cells and in these instances, reverse the fibrosis in the liver. I said I would just mention a very important topic today, which is the exploration of other sources for CAR therapy. Today's therapy chiefly makes use of autologous T cells that are collected from the patient himself or herself. But could we go to other alternative sources that would allow us to not have to produce the T cell for each patient? That would require making them from one source and infusing them to a multiplicity of different recipients. Well, there are approaches underway. These are early days. There are clearly a number of challenges to overcome. This is a cell, not just a molecule. And while this is not a transplant, it is still infusing the cell from one individual to another. Again, a number of us are hopeful that the tools that we have today for gene editing in particular will allow us to generate allogeneic T cells. And one approach that I want to mention in particular that we are particularly interested in is the ability to potentially generate T cells from pluripotent stem cells. In this instance, were we to succeed in doing so, the T cells now would not be born in the thymus, as I said at the very beginning, but they would be born in a bioreactor. And so a question is, ultimately, someday, will it be possible to engineer not just the CAR, but the CAR T cell or the CAR NK cell on plastic in culture? So to finish, I just want to put this CD19 CAR therapy in a broader context. Cell therapies have existed for several decades. I mentioned earlier LAC cells, tumor infiltrating lymphocytes, donor leukocyte infusion, and virus-specific T cells. But in all of these instances, the investigators had to painstakingly look for good T cells, either in the patient or in a donor. What the CD19 CAR paradigm shows is that it is possible to engineer T cells and dictate what antigen they will recognize and also what functions they will acquire. And so the new paradigm that emerges is that we can manufacture T cells using gene transfer or gene editing, physiological receptors, or chimeric receptors, and further synthetic biology tools to reprogram and rewire these T cells. And so the main message would be, if you cannot make a T cell response to the tumor, or if you do not have the time to make that response in vivo in the body, well, don't worry, we'll make it for you. And yet at a higher level, I think that this approach provides a third means to master, if you like, T cells in medicine. The first, and for sure the most important, is active immunization, which we hear about every day today, talking about the COVID vaccines. The second, already mentioned, is the release of latent but ineffective responses in some cancer patients. And the third one, illustrated by CAR T-cell engineering, is a synthetic form of immunity, in which synthetic or normal receptors are used to instruct supraphysiological immunity. I'll stop here, just leaving the next slide up, showing lab members and many visitors to the labs today, and show you again my long-term collaborators here, Isabelle Rivière, assisted by Xian Wang, who manufacture all these CAR T cells now for over 400 patients that we have treated at Memorial Sloan-Kettering. My former fellow, Rainier Brentianson, now head of our cell therapy service, and Prasada Dusumili, who runs our solid tumor program. I thank you for your attention.
Thank you very much, Michel, for a really great talk. I think you covered a lot of ground in a short period of time. Thank you. We have some questions from the audience, and I guess I have a couple of questions as well, but I'll start with the audience. Well, the first question, actually, you covered to a degree, but you talked about why there is a variation between patients in their response to CAR T therapy. And the questioner asked whether some of that could relate to underlying genetic differences between people. In other words, is there a genetic component to the response to CAR Ts?
Yes. That's a great question. It is perhaps the biggest question at the end. Why does it work in some instances and not others? This is a very complex situation to analyze, to deconstruct, actually. Unlike the monogenic disorders that Kathy will tell us about in a few minutes, where there is, one gene, monogenic disorder, one gene that is lacking or defective. In this case, the cancer is never identical between two patients. The number of mutations are different. And of course, the genetics of the patients are different. Their age is different. Their prior therapies can impact on the quality of T cells, and of course, their age. So we're dealing with a far greater number of variables. Nonetheless, it is essential that we tackle this question. Overall, I'm not aware of a genetic variation, a germline variation, that we could, at this time, use to predict greater efficacy or lesser efficacy. Having said that, a lot of work is being done at this time in defining what T cells in particular are the best vehicle or the best substrate, if you like, for this therapy. And in doing so, it is possible that we will eventually uncover why some patients are more prone or more likely to develop an effective response compared to others. But I don't think that we are ready to just answer that question at this time.
Okay, thank you. A couple of questions sort of related more around access and cost and so on and so forth. One is you mentioned, of course, that most of the trials going on today are in North America and China, Europe. So the therapies are very expensive at this point. How do you see it going as we try to move this out across the world? It's a challenge within our current healthcare systems to get these expensive therapies into the clinic. But how can we make sure that we get global access to these therapies?
Yeah, that's a great question. It's a great question. I have to first emphasize how innovative, and some would say revolutionary, perhaps, this approach is. That's a strong word, but it might apply here. This medicine is not another chemical or small molecule. It's not a recombinant protein. It's not an antibody or a clotting factor. These chemicals or these proteins are drugs or medicines that the pharmaceutical industry has been producing for decades, and where the methods of manufacturing and distribution are well advanced. But this therapy is a cell, and to boot, it's an engineered cell. And so this is very different. And so all of this formidable experience of manufacturing does not exist here. This therapy is born out of a few academic centers and was actually very rapidly transferred to pharmaceutical companies, but those did not yet have the time to really optimize the manufacturing. And by that, I mean to miniaturize, to robotize, to accelerate the production. So that effort is only starting now. So I would say that, therefore, this situation requires a bit of patience, where this is ahead of what the pharmaceutical industry can produce at the present. Having said that, it's amazing to see the enormous enthusiasm that has come forth in the last years. People from the aeronautical industry, acoustic industries, engineers with all kinds of backgrounds being drawn now to perfecting, if you like, the culture and production of cells. I won't address the price per se. It's not my expertise. I'll just say that I think for people like myself, it was with sadness, really, that we saw these very high costs, or these very high prices for the first CAR T cells. All I can say is that that's beyond the control of the scientists. It's really the private sector, the regulators, the insurance government who will set an adequate price. What scientists like myself can do, though, is to improve the quality of these T cells, better understand what makes a good T cell. One simple principle is if there are better cells, you'll need fewer, and make an economy of cost of goods just from that perspective. And so what we think we can contribute really is making a better T cell and also exploring these allogeneic approaches, including these pluripotent stem cell based approaches that may then result in having these vast reservoirs in bioreactors, churning out T cells for their adoption, for their use on a larger scale. I want to lastly, I know I'm taking lots of time, but it's a huge question, share with you a comment that I heard that I found very interesting at a meeting at St. Jude Research Hospital. Someone pointed out that the use of CAR T cells could be particularly effective, useful in poorer nations. And the conversation was about treating leukemia in Africa. And the point that was made is the following, is that patients come sometimes from far away to a big center. They get an induction chemotherapy, then they go back home and they have to continue their treatment. And often the treatment, there's poor compliance or the drugs are not available. And when you look at the results, indeed, the treatment of leukemia in poorer nations is dismal compared to what it is in our wealthier nations. And so that expert pointed out, wouldn't it be great if you had a CAR T cell? You just come to the hospital, it's a one-time treatment, you get it, and you go back home, and there is no additional treatment. And I was kind of startled to see someone thinking globally about one of the very attractive features of a living drug that you only give once.
Thank you. That's a great point to end our discussion. I think that's extremely hopeful for the future. And I guess in that regard, of course, we've seen the Gates Foundation, for example, working with NIH to develop gene editing approaches for genetic diseases to make them available globally. Maybe they also should get involved with living cell therapies and thinking about how to turn them into a global tool. So thank you very much indeed.